Okay, hi everybody. Thanks for joining us. Welcome to Sigma Xi's series of Google Hangouts with its distinguished lecturers. I'm Fenella Saunders. I'm the managing editor of American Scientist, and this particular hangout is sponsored by the RTP chapter of Sigma Xi, so hi to all the members. Today we are really fortunate to have an hour with Omowunmi Sadiq, who does research on biosensors, and she's going to talk to us today specifically about some work related to detecting food pathogens, but we'll also talk about some other areas that she's gotten into. So thank you so much, Dr. Sadiq, for coming.
Thank you for inviting me.
Great. So let's just start out with a little bit of the basics and talk about, can we define what a biosensor is for people who aren't familiar with them?
A biosensor is a small size device that has a biological component in close connection with a transducer. And typically, a biosensor could be used to detect some biomarkers in the body or in any other kind of sample, in foods, in air, in water. A very common biosensor that people might be familiar with is the glucose biosensor. Typically, these are used by diabetic patients to check the glucose level in their blood. So most people are probably familiar with patients pricking their finger nails and getting a few drops of blood and checking how much glucose they have. Essentially, that is a biosensor. It has a very small amount of an enzyme called glucose oxidase, and the glucose oxidase essentially converts glucose in the blood to gluconolactone, and it gives patients immediate information. And so one can potentially design a biosensor for anything you want, as long as you can come up with the transducer as well as the biochemistry to make it work.
Okay, so as far as I understand it, one of the advantages of this kind of technology is being able to use a very small sample and sort of amplifying either it or the signals. Can you talk about that aspect of biosensors?
Yeah, biosensors in general don't need a lot of samples. As I gave the example of the glucose biosensor, all one needs in most cases is just a few drops of the sample, and it has the ability to amplify the signals and also give you immediate information.
Right, okay. So can we talk about some of the other aspects of what you're doing here in terms of the food pathogens that you're using the biosensors to detect? Maybe we can give people a little bit of context of how big a problem foodborne pathogens are.
Um, Food pathogens, first to define them, are either bacteria or viruses or even fungi that one can find. They're everywhere. However, some of them are really deadly, and from time to time, they do find their ways into the food. And viewers might probably be familiar with uh, the Center for Disease Control, uh, you know, giving some information about certain food recalls. And so, most common would probably be E. coli in spinach or in foods, in milk, in drinks. And so, in an ideal world, nobody really wants to have the food that they're eating to be contaminated, but it does happen, and it happens more frequently than we like to admit. And so, there's always the need to ensure the safety of foods.
So just to clarify though, the organisms that we're talking about, not all of them are harmful under all circumstances. I mean, some of them we need in certain roles, so it's more sort of a contextual problem when they become foodborne pathogens. Is that true?
That's absolutely correct. In fact, we have bacteria all over. We have them on our skin, we have them in the air. We need, in fact, E. coli, for example, we do have in the gut. However, the ones that we're talking about are those that are pathogenic, which means that disease, causing, they will cause disease, they make people sick.
Right, okay. So there have been ways to detect these organisms in the past. There's some established methods, but I wondered if you could talk about some of the problems with more traditional detection methods, such as culturing or other methods like that. Is it just that they're slow, or are there other problems that you're trying to get around with using a different kind of sensor?
So there are established methods to detect food pathogens. The most common would be the plating culture, to culture the pathogens in the lab. And this usually takes between 48 to 72 hours. And in most cases, the time is of the essence when you really want to ascertain the safety of foods. And so people really don't have access to these things. And also to think about how much, many times we do process large amount of food. And so how would you then be able to see, to ensure the safety of massive, volume? And so you need on time, in situ, immediate information about the safety of foods. And so this is where biosensor really comes handy.
Mm-hmm. Are the biosensors able to detect a lower level of pathogen than traditional methods can?
Yes, the biosensor are able to detect lower levels. Uh, in fact, there is a current USDA challenge to see how much, to actually detect a single bacterium in like 100 mils of solution. This is very, very, very small. And existing methods cannot provide that kind of information. And so biosensors, especially when they're linked with nanoparticles, uh, enhance the sensitivity. And so they're able to go to very low levels of those pathogens.
Mm-hmm. Are the biosensors also potentially less expensive to process?
Biosensors are, in fact, their advantage is the low cost. The low cost in terms of fabrication. Our biosensors are made from papers, so the papers are cheap, and we don't use extensive electron fabrication, photolithography design, which also makes them very, very cheap.
Right, okay. Um, and with the fact that you can get them sort of more widely spread into the food system, is it then possible that they could enable earlier detection of a problem or an outbreak?
They will enable earlier detection, and they are also capable of, we can, because they're cheap, we can create a whole lot of these things and strategically place them out in environment where uh, it's, food testing is easier and they're very immediate. Anybody can use them. You don't have to be a specialist to operate and take, just like home pregnancy kits, just like the diabetic patients can monitor themselves. The concept of a food borne biosensor is the same.
Right, okay. So let's talk about some of the mechanisms that you're actually using here to do the detection within the sensor. One of the things I think you use is something called electrochemistry. I wondered if you could explain that mechanism, please.
So in electrochemistry, we simply we're using the ability of we're looking at the biosensor itself, the pathogens themselves can metabolize. And so as they grow, they use oxygen. And the oxygen can get reduced, and we monitor water. And so basically that's what the electrochemical principle is doing, is using the intrinsic ability of the pathogens to use oxygen. And so, because electrochemistry allows you to monitor the oxidation reduction of oxygen, and so we see that as the oxygen peak is going down, we know that the bacteria is increasing in number, and also it also tells you something about the viability. That is, can they make people sick? It's not enough for you to know whether or not they're there, but can you tell whether there's still it's possible to make people sick? Are they viable?
Right. Okay. So, so what's actually happening is you're saying the oxygen level is going down, and that is changing the electrical signal that you're getting out of the sensor. Is that right?
That's correct.
Okay. So, like the level of the signal that you're getting reduces with the level of oxygen.
That's right.
Okay. So I think that you were saying just now, what to sort of add on to that is if you're saying it's not enough to just know they're there, you kind of want to know more about them. Can you sort of create an electrochemical fingerprint for specific pathogens or levels of pathogen?
In fact, that's what we're doing. We're creating specific fingerprints for different types of pathogens. And we're also using the intensity of the signals that we get to tell us how much pathogens are present.
Mm-hmm.
So we have created a whole database for different types of pathogens, and so we know, the specific fingerprints, and we use that. We link that to software models to train the sensor. So the sensor now can tell you if that E. coli is there or if it's salmonella. And we can also tell you with some levels of accuracy what percentage confidence we have. Ideally, we would like to have close to 100% accuracy, but right now we're close to 90, 95 plus percent.
So you're actually be able to tell different types of pathogens that are there. Does that have to do with the differing metabolism of the individual organisms?
That's right. Yes. So they metabolize differently, and so the fingerprints are different in terms of their electrochemistry, and that provides information about the type of pathogen, and it also provides information about the quantity, the amount, the number of the pathogens present.
And how long does that kind of measurement take? How long do you have to watch the metabolism change the level of oxygen before you can kind of get a full fingerprint for an individual strain?
Yes. So ideally, for the electrochemical sensor, biosensor that we developed, we would get information within the first 1 hour. If you compare for the viability, because you need to be able to monitor the oxygen. But if you compare that with conventional methods where you need 48, 72 hours, that is a great time saving.
So you can tell within an hour what strain it is, or just that there's something there?
Within an hour, we know what strain it is, and within an hour, we can tell if it's viable.
Okay. And so this work sort of seems to really cut across a lot of different disciplines. Can you talk about the importance of that? I mean, you talked about a sort of a almost a computer. You're training sort of a sensor network. So can you talk about combining all those different areas?
That's correct, because biosensor, in fact, sensor in general as a field, is multi-disciplinary. You do have people, we do work with chemists, physicists, we have material scientists who design the electrodes, we have engineers who actually fabricate the device for us and do the testing. So it tends to be multidisciplinary, and that's one of the advantages of our center. In our center, we do have, the cross-pollination of all of these different disciplines working together.
Right, okay. I know you also work on some custom polymers. Are those used in this kind of sensor?
Yes, our custom polymers are fabricated within the sensor strips. So because if you consider that we're using paper as a template, and so if you put paper in water, it's not going to hold up. And so the membranes are used to coat the paper and make them more durable, and they can be used in liquid samples, in water samples, and they hold up.
Are those polymers also used in this process of creating that electrical signal out based on the oxygen level that's in the sample?
Absolutely, the membranes are conducting. They are conducting polymers, and so once we coat the papers with the membrane, they help with the electrochemical signals.
So as I understand it, another detection mechanism you're working on is using the same kinds of carbohydrates that pathogens are actually using to get inside cells as a way to detect them. Can you talk about both of those aspects? How does a pathogen use a carbohydrate, and how do you actually use that as a detector?
So we're simply using the same mechanism that the pathogens employ to gain entry into the human body. For example, E. coli has pili, and on the pili you have different types of proteins, and those proteins are specific for attaching onto the cell. And so they basically use the carbohydrates to stick. They basically stick onto the body. And so what we're doing is creating molecules that would allow the ligands to have effective uh, specific recognition onto the bacteria. And so, and we found them to be very, very effective actually,
Mhm.
than simply using try and error, to gain entry. So we're basically using the same mechanism that the microbes are using to detect them.
So are the microbes kind of using the carbohydrates almost like a glue to stick to the surface, or is there like a receptor involved?
There is a receptor involved, but it essentially the carbohydrates are more or less the glue with which this happens.
And so are you engineering a carbohydrate to kind of attach to specific microbes, and that's how you're detecting them?
Yes.
Okay. So are these are ones that are actually being developed in your laboratory? You're sort of engineering them to work with the same receptors that the microbes have, or how does that work?
Uh, we've done this successfully for E. coli, for different species and substrains of E. coli. And so the way this works is that for I don't want to go into the specifics of the molecules that we're using, but essentially you have the carbohydrate linked onto the uh, like a sulfur derivative with gold onto our electrode. And so the carbohydrates, this attached to these long ligands, are able to bind onto the surface of the cells. And so they glue onto it with high specificity, and that gives them the recognition. And the other advantage of that is that because you're using the specificity of the reaction, it's not likely to recognize any other molecule that is not specific to it. And so it's a great advantage. So if we've designed the ligands for E. coli 0157, it's not going to be recognized by Salmonella.
So that way you know specifically what you have if you get a signal out, basically.
Mm, yes.
And if you wanted to make a more general sensor, could you combine several of these ligands within, say, one sensor sample?
Yes, if we want to make it general, then we create arrays of this. So we have arrays of the different ligands. And so we can detect more different pathogens.
So, what kind of real food samples have you successfully tested with these kinds of sensors so far?
So far, we've looked at sushi, we've looked at spinach, we've looked at water, We've looked at juices, apple juice, different things like that.
Hmm. And do you think that this sensor would have use in the developing world as well as in, the United States and other countries like that? Is it cheap enough that it could be used in places that have less infrastructure, for instance?
And that's one of the advantages. That's one of the goals, that the sensors will be so cheap enough that um, they can be used in places with less resources, um, and also with not much infrastructure. So you don't need extensive plate readers to give you the answer. You either use your handheld cell phone. Everybody has cell phone now, or you simply use the visual color change to give you an idea of what's going on.
So let's go back to that point for just a second. How do we connect this back to sort of everyday personal devices such as cell phones? Is there a way for people to be creating readouts that they can interpret with their own devices?
That's the subject of our research.
Right. Well, that's the goal, obviously, but how would you go about doing that?
Oh well, we're writing apps to uh, translate the color change into signals to let people know what's going on.
So how would you, so the phone would look at the color of the sensor and be able to have a database to compare it to?
Yes.
Right, okay. Um, is there also a possibility of the phone doing any kind of further interpretation of the results? Would it need to like have a spectroscopy program or something like that, or is that getting too complicated?
No, we want it to be very simple. It should be such that the user can tell you basically what they have, yes or no, uh, color or no color.
Right,
So it should be very simple, low tech uh, idea.
right. So you wouldn't have to necessarily physically connect the sensor to the phone to do the readout?
Yeah.
Okay. Um,
But we have a device that goes in, just like you would put the earplugs into your cell phone. So we have a device that does that.
what does that device do when you plug it in?
It translates the signal from the sensor into an image on the cell phone.
Is that for the electrochemical sensor, or is that for the other kind of sensors that you do?
It is primarily for the electrochemical. However, we still find a way to uh, convert the electrochemical signals into some color uh, information.
Right, okay. Um, excuse me. We've had a question from the audience asking about the challenge faced with local food sellers who may be unscrupulous and sell tainted food anyway. If you had any ideas about getting around those issues?
Can you take that question again?
Excuse me. Sorry. There's a question that asks if you've thought about local food sellers who might be trying to sell tainted food anyway, and how you might get around that problem.
So local food sellers are trying to sell, you mean contaminated food?
Right, right,
Absolutely, because in fact, that's one of the ideas is that, you know, people, first of all, the individuals themselves should be able to tell. Uh, and the device should be low cost enough. However, local food sellers can use the technology to assess the safety of their own food, to be able to know when to not to sell it. If it's not fresh, if it's going bad, even though it still looks visibly okay, but the sensor will tell you based on the number of pathogen, going up, that it's probably not safe to eat. And so, food sellers is a good. It would be a good technology for them to ascertain the freshness of foods.
Right, but I think the part of your research is also getting the sensors into the hands of individuals so you can test food you're eating at a restaurant or even at
Right.
an end user level. Is that
That's
right?
correct. That's correct. Yes.
So what are sort of some of the next stages for your research? How are you going to try to develop these into something of more of a consumer product or anything like that?
Um, Currently, we're trying to make we're creating a gizmo, if you like, to connect our sensor onto the cell phone and to make that really cheap. Right now, we're not being cheap as we would like to be. And secondly, we are working on developing very simple app that will make people understand the information that they're getting from the sensor.
Just so that, again, it goes back to this sort of cross-disciplinary approach you're taking. So you're writing apps as well as developing custom chemicals.
That's correct.
You must have a very diverse lab group.
That is one of the uniqueness of our center. It's a center. It's called the Center for Advanced Sensors and Environmental Systems. And in our center, we do have very diverse fields working on the problem. On this particular project, we have, let's see, we have my lab in the chemistry. We do have a biomedical engineering group. We have a electrical engineering group that's involved in this. Yes.
Mhm. So I'd also like to ask you to talk a little bit more about other applications of sensors like these, because I think that you're looking at a number of different areas, everything from cancer to pollution. So I wondered if you could talk a little bit more about those sorts of connections.
Right. Going back to my earlier example with the diabetic patients, um, biosensors could uh, be used in a broad range of applications, uh, ranging from biomedical to point of contact patient applications. We have sensors for detecting the level of pain that people are going through. We call that the pain biosensors. We also have sensors for environmental toxins, metals, heavy metals like chromium, lead, um, in water, in soil, in air. So we do have sensors for different applications.
Do those use the same kinds of polymers that you've developed? Is it the same kind of sensor, or are there different mechanisms that you're using?
No, these are different mechanisms. The pain biosensor actually uses fluorescence principle, and it rather than using the strips for the food, it uses capillaries. And the capillaries, it uses flow. So we have to have a flow. We can use the flow blood, very small amount of blood or liquid samples through the capillary. And the sensor itself is contained within this tiny glass chip that has unique recognition chemistries worked into them. And those are being used to look at the protein biomarkers in blood so that we can now correlate the level of the protein biomarkers to the level of pain that people report on their chart when they come into the hospital. So those are essentially very different principles and even different templates.
So how are you actually applying those to a patient? Are you going to have to put them inside a vessel, or how does that work?
Right now, we're still doing the same thing like the diabetic patients. We take few drops of blood. In fact, we've looked at well over 200, 300 blood samples from people who come in for different kinds of operations in the hospital. And yeah, so we still need the blood. We need the blood samples.
Hmm. And so when you're doing, for instance, cancer detection, is that a similar mechanism?
For cancer detection, one will be looking at either looking at it from the genetic standpoint or looking at it from the protein level standpoint, and or even cell, to look at the cell level. So, so for cancer detection, mostly we've looked at cancer cells, non-cancer cells. We compare uh, signals from different kinds of cell lines.
Hmm. And so I think you've also had some success with an HIV sensor. Is that correct?
Yes, we've looked at HIV antibodies in the past. Yes.
And is that the same kind of sensor if you are using a blood sample there?
Blood samples or other forms of biological samples from the blood, from the body.
Hmm. And is the advantage there again the speed of the result?
Again, the speed of the result as well as the cost, and also the accuracy as well. You really want to be very accurate with things like that.
Is that something that's now in clinical use?
No, not at all, because the process of getting that to that stage is, it's quite involved.
Hmm. So you're still in the experimental stage with that?
Yes.
Okay. Um, we've had a question from the audience asking if you could talk about some of the ethical considerations involved in working with nanotechnology. Um, there are there's some controversy now about safety with nanoparticles in the environment and things like that. So I wondered if you might address that.
Absolutely. One of the uniqueness of nano materials is the fact that they have very high signal to surface area ratio, and which implies that most of the electrons are in the surface, on the surface. So they have larger percentage of their electrons in the surface. This makes them very reactive. It makes them uh, exhibit remarkable characteristics. Let's take gold, for example. Gold is a noble metal. It's not very reactive. That's why we can use it in jewelry. It's stable. Gold, conductivity of gold will not change. Uh, However, at the nanoscale level, uh, gold is always like kind of yellowish, shiny, ductile, malleable. Is now giving you different colors. It could be red, it could be purple, it could be blue, depending on the particle size. Now, you can utilize those advantages for sensing, enhancing sensing. You can use that for, maybe energy materials. You can use them in nanomedicine. But then it also raises the question of ethics. What happens once we expose people or the environment to these very reactive materials? What are the likelihood of unintended consequences of these things? And so those questions have been coming up. And in fact, I have been involved in starting an organization called the Sustainable Nanotechnology Organization that tries to address to educate the public and to try to propose that we develop nanomaterials, both the application as well as the implication concurrently. We don't want to wait until 10 years, 15 years to discover that uh, these are the negative impact of some of the materials that we're using and exposing people to. And so, yes, the ethical questions are there, but scientists and engineers are actively looking at this and trying to understand the fate, the transport, the transformation of nanoparticles in the environment. So, yeah.
So there's a lot of controversy right now over, I think, silver nanoparticles, which are used in cosmetics and things like that. So I think this is a very public issue at the moment. I'm not sure that anybody knows definitely that silver nanoparticles are causing issues, but they have been found in places where they weren't intended to be. So I think transport is one of the bigger issues here as well.
Right. Yeah, silver nanoparticles. I mean, if you look at silver, generally, it's been used for a long time, even before we start making, some of the nanoparticles. They are antibacterial. They have antimicrobial properties. They the challenge is now is what happens when we infuse silver nanoparticles in consumer products. And those consumer products are now finding their ways into, people wash, you can buy nano socks in Walmart right now. You buy socks that's been infused with nanoparticles, they perform the function for which they were designed, which is to kill the bacteria. But then you wash the socks, you put it in your laundry machine, and more than 80% or even 90% of the nanoparticles get washed off. Everything will find its way into the wastewater system. And so that is where the problem lies. How do we keep track of such exposures? What are the likelihood of people getting exposed to some of these uh, particles that we don't want them to be exposed to, that's very reactive? And how would that affect the microbial population in the environment? Is it changing that? Is it not? Uh, we still need a lot of research to better understand what's going on in this regard.
So do you think that some of the regulatory agencies are scrambling in a sense to catch up to the nanotechnology aspects of this, because a lot of these materials are been deemed to be perfectly safe at macro scales, and there's no rulings for nanoscale level sizes of these particles? Is that something that's being addressed on a regulatory level?
On regulatory level, I think the EPA, for example, is funding research to better understand the uh, implications of nanoparticles in the environment. There are regulatory agencies that are kind of, again, not, I wouldn't say playing catch up. I think they've been there because it's unlike in the past with things like PCBs or mercury, where we suddenly find, they're not desirable at certain levels in the environment. People raised this question very early on with nanoparticles. And so where they're concurrently looking at both applications and implications. So the regulatory agencies are there. Um, in fact, you can really follow up on the status of some of the particles. OSHA is working on this for exposure in the workplace. And so I think there's a lot of work. We're looking at the applications and implications concurrently. So it's not, I don't think it's a matter of catch up, but we're more aware of these things. It's not like in the past.
Right, okay. We've had another question, which is asking, in the US, the FDA will need to approve a product that's used for food certification. Do you have plans for pursuing this line to get, I guess, the regulatory approval for the food pathogenic sensors, I guess is the question.
Well, certainly it would have to be approved. And part of what we do is the research and development. The mechanism for approval would depend on companies that would probably license the patent from us, and they would have to go through that process. But for us as researchers, we don't get involved with that. But for the companies, they will have to do that.
So you had mentioned that a lot of these things are still experimental, obviously, but do you have sort of a potential timeline for when these could be ready to be actually be used in a more consumer application?
I think the food pathogen sensors are more closer to real applications than the pain biosensors, for example. So we're talking about 3 to 5 years. I think that that would be doable.
And for the more experimental ones, such as the cancer sensors or the pain sensors, you think that's more a decade out or something like
Yeah,
that?
those would be more because I mean you have to anything that you have to yeah, that would be longer. I
Mhm.
can see those would be much longer.
Right. And for environmental sensors, if for things that are sensing heavy metals in the environment or anything to do with pollutants, would those be on a similar time frame as the food pathogen detectors, or are those similar technologies?
Yeah, those would be comparable to the food pathogen sensors. Yes.
Hmm.
Yeah,
I think that your work could potentially also be used in areas even like detecting explosives, if I understand that right. Is that correct?
that's correct. We've worked with Naval Research Lab in the past on a similar on that kind of project. Yes.
Okay. So I wonder if we could switch gears just for the last few minutes that we've got here. And I wondered if you could talk about some of your background and what got you interested in doing science, what it was like to be learning science where you grew up, and to just talk a little bit about that.
Yes. I grew up in Lagos, Nigeria, and I was very fortunate to have parents who encouraged me into the sciences and emphasized the you know didn't make me feel like you're a woman, you're going to be a girl, you don't need to study math or science. So I have siblings. Three of my siblings are in the medical field. They're MDs, and so I was very fortunate to have a very structured background, disciplined background as well, that allowed me to really focus on my studies. Yeah.
Did you encounter any stereotyping during your school years that you found discouraging, or did people try to tell you that you shouldn't be a scientist?
Uh, Interestingly, while growing up, no, because I remember at the University of Lagos, I had two female professors in chemistry, and so it wasn't an issue. But since living in Nigeria, I think I've seen that it's not as. Of course, I've experienced that since living. Yes.
Hmm. And do you have any advice for people who may encounter situations like that, how they can handle those kinds of stereotyping and not be sort of pigeonholed?
I would say that you know you should focus on what you want to do and don't listen to detractors. If you find that you have the passion, you have the penchant for science, for research, I think you should pursue your goals regardless of the stereotypes. Um, you probably will find that you may need to work harder, maybe two times more to be taken seriously. You, but I would say go for it.
So, when you were growing up, I was reading that in one of your schools, I guess when you were younger, you were actually using candles for light while you were in boarding school, and
Right.
now you're doing very high-tech work. So it's sort of an interesting dichotomy. I wonder if you can comment on that. Does that feel interesting, or is that something you never would have imagined when you were younger, for instance?
Um, I didn't even look at it from as a disadvantage, believe it or not. I the the incident that I recorded in my autobiography was more of light out, where you're in a boarding school environment. It's a very structured environment, and you have to turn off the light. You need to go for prep class in the evenings, and there was no light in the hall, and you had to use lantern. And of course, I was even trying to do experiment with that. I didn't look at it from that perspective. I looked at it as a trying to overcome challenges and trying to be stronger and not be deterred by your environment. And that is what I have tried to imbibe in my children. That you, even though you're growing up in a more in a different environment, but still you still have to overcome your own challenges. You have to overcome the stereotypes. You have to overcome the limits that the society places on you, by not giving up, by simply taking things like, you know what, you run with it. Don't let people put you in a box, and don't let that stop you from what you want to do. The only person that can stop you is you.
Right. I'm sure that you have students that you advise or that you teach that come from many different backgrounds. And I wondered if you could talk about how you might advise students who come from less advantaged backgrounds to make their way in science.
I will advise them to uh, note that they'll probably be the minority in their environments, and uh, and it may not be unusual even for instructors to look at you like, what are you doing here? You don't belong. But to look at all of that and just ignore it, and determine that no matter what, they're going to be the best, and they're going to be the best. And it takes hard work, it takes a lot of hard work, and it's merit, and you have to prove that. So it's not what people say, it's what you believe yourself to be. If they think you can do it, prove them wrong. But yes, you can.
So, one of the topics that often comes up when talking about students in disadvantaged backgrounds, perhaps ones who have been the first generation to go to college, has been one of the things that they face is that they have no network to lean on. They don't have the same kind of intrinsic network that other students might have. Can you talk about mentoring students or ways that that can be done to help those students feel like they have more of a resource network to tap into?
I do. I've mentored a whole, range of students of all backgrounds, uh, disadvantaged, underrepresented, and everybody. Uh, Everyone has, it doesn't matter who you are. We all have one thing or the other that we have to overcome. But what I try to tell them is, try to find, others of like mind that you can rub shoulders with, that would help you, uh, you know, you know, at least discuss what you're going through. You realize that you're not the only one. You're not the only one, and everybody has one thing or the other that they have to overcome. Whether or not they accept, whether or not they agree with it, whether or not they're even aware of it. So try not to be a lone wolf. Try to identify other people of like minds, that that you can encourage one another, and create a community of like minds.
Yeah, of course. And I just also wondered if you could address the question of women in science and the academic process. There's a lot of talk about women in tenure track positions, the difficulty of merging that with having children and families. I wondered if you could talk about your own experience in that area and if you sort of have any takeaways from that process.
Uh, yes, it's a challenge, because you have to be everything. You want to do research, you're going to be, you're going to teach, you're going to do service. There's an inordinate amount of time required to be on committees and things like that. And it's not, I wouldn't say, I say it's challenging. You have unique challenges. However, with good planning, um, you can still achieve your goals.
So we have another question from the audience asking about the fact they says that you have a number of patents for your work on biosensors. So do you have any words of wisdom or encouragement for those going through the patenting process?
Uh, the patenting process is a very patient one. Sometimes, you the description or even the documentation of a new idea or innovation or invention um, is different from traditional peer-reviewed publications, and it can go back and forth with with the examiners. I would say it needs a lot of patience, and sometimes even the going back and forth helps you to fine tune the ideas. It helps you to even explain things a little better. But I would say that with a good intellectual property office and your lab and or your research, I think you will make success of it.
So everything that you're talking about makes it sound like one of your strongest skills is time management. There's so many pieces to everything that you do. I'm wondering how you fit it all in.
It's not, I'm still trying to figure that out.
Yeah, it seems like, you've got to worry about so many aspects of things, even if you have assistants and staff and things like that. It just seems like all the pieces, keeping them straight, must be a job in and of itself.
Yeah.
Um, so the question there, I guess, might be, for people who are younger, who are looking to get sort of leadership experience that might lead them to be better lab managers and to manage students and things like that. Do you have any tips in that area for students who are trying to sort of gain some experience in that area?
I think a lot of it is, time management is critical. It's very important to manage the time, what you want to achieve, in a typical day, block times for, different things, because there are things that would always, demand, and everything is rushing for your attention. Everybody wants a piece of you. So priority, you have to prioritize. What do I really want to achieve? And you cannot be everything to everybody. You're not likely to do everything, but whatever you want to do, focus on that and make sure you do it well.
Okay, great. Well, we're just about out of time, so I'm just going to wrap up here and say thank you very much to Dr. Sadiq for spending an hour with us and for sharing your insights not only on your research, but also on the process of science in general, which is getting more complicated all the time. And I just want to thank everybody for viewing. If you have follow-up questions, you can post them on Sigma Xi's communities page called The Lab, and we'll get them to Dr. Sadiq, and so she can answer them. And hopefully you'll join in the discussion there. You can also check that page and our Google Plus page for upcoming chats with other distinguished lecturers. Our next one will be October 15th with Krishna Foster, who's going to talk about air pollutants. So thank you very much, Dr. Sadiq, for joining us.
Thank you so much for doing this. Thank you so much.
We appreciate your time very much. Thank you again. And if chapters are interested in having Dr. Sadiq visit them as a distinguished lecturer, you can check the distinguished lecturer page on the Sigma Xi website to see details how to do that. So thank you very much again, Dr. Sadiq.
Thanks a lot. Thanks.